Hello, everybody. This is Scott Roberts. I see Chris Larson is our first log on, or first chat anyways. Cameron Gillis and Book Davies. Yep, it's Friday. And my fascination turned to DNA today. So, and where did it, what were the building blocks for DNA? How did it get here? That's something of intense interest to astrobiologists. And, one argument is that it may have arrived in asteroids or comets or meteorites, or maybe all three, mixed together with all the rest of the organics here on Earth. So, but it's interesting to know that they have found some of the basis for DNA in meteorite samples. So I think it's going to be, there's a little video here from NASA astrobiology that will talk a little bit about it. Israel Monteroso Villatora. Israel, where are you from? And Tim Myers, Andrew Corkill is here with us. Book Davies. Yep, nice to see all you guys.
Well, I know my 9407.
Oh, wow. Asteroid 3 Juno passing close to Messier 10 globular cluster tonight. Good tip, Mike. Guatemala. Yeah. Nice to have you watch with us today, Israel. That's cool. Israel, that's cool. Martin Eastburn, you made it. Yes, you did. Pekka got too happy SMS today from Explore Scientific as sender. Okay. That's cool.
My name is Mike Callahan. I'm a research physical scientist in the Astrobiology Analytical Laboratory at NASA Goddard Space Flight Center. We've discovered a variety of nucleobases and nucleobase analogs in meteorites. And what a nucleobase is it's a small molecule that's found in DNA and RNA, and these molecules are essential for all of life. And so our results show that these molecules are extraterrestrial in origin and the products of a chemical reaction occurring on the meteorite. We believe that these nucleobases and meteorites are extraterrestrial based on 3 reasons. And the first is that we find nucleobase analogs in meteorites. What a nucleobase analog is that it is a molecule that is structurally similar to the nucleobases you find in biology, but it's different. And that these structures are actually either rare or even absent on Earth. The second reason is that in addition to looking at meteorite samples, we also study terrestrial samples. So these meteorites are collected mostly in Antarctica, and one that's very famous called Murchison was collected in Australia. So we have soil samples from that Murchison area in Australia, and we've also had, we were lucky enough to get an ice sample from Antarctica where some of these meteorites were collected. And so when we look at these soil and ice samples in the laboratory, we don't see the same distribution of nucleobases. And with these nucleobase analogs, we don't see them at all in these terrestrial samples. The third reason why we think these nucleobases are extraterrestrial in meteorites is that in the laboratory, we study reactions of hydrogen cyanide. And when we extract these, the products from these hydrogen cyanide reactions, we also get nucleobases. And we actually get a similar suite of nucleobases to what's found in the meteorite. And so when we saw that, we were really excited because we were thinking, well, hydrogen cyanide is dispersed everywhere in the interstellar medium, and it's likely to have some sort of chemical reaction on the meteorite. And so when we saw that, that really convinced us a lot that we're seeing something from a chemical reaction rather than some sort of biological contamination coming into the meteorite. So this has implications for the origin of life on Earth. We know that meteorites contain amino acids, which are the building blocks of your proteins, and now from our research, we can show that nucleobases, which are the building blocks of your genetic material, like DNA and RNA, are also found in meteorites. And so these things together could have seeded an early Earth with these really important molecules that could have built up to the larger molecules you see today that are essential for biology.
Hey everybody, it's Scott Roberts here, and this is my 8th MicroCosmos Voyages. And the reason why I picked out MicroCosmos is that I think of diving into the microscopic world is just, everything's connected to the universe, right? So it's just another way to explore the universe in that way. The video that we just watched, shows a researcher, an astrobiologist, who is finding some of the nucleobases for that could have created modern day DNA. So they're still trying to work that out. But I think for over half a century, there was a big argument about whether or not that these nucleobases bases from meteorites could have played a role in forming DNA as we know it today. I think that some of the latest findings, though, are pretty compelling. So pretty interesting. So it made me think about, well, gosh, would it be possible to see DNA? You notice that they were like crushing up, pulverizing, I think it was a carbonaceous chondrite meteorite, and to search for these nucleobases that are found in some of these meteorites. So I found out it wasn't as hard to separate DNA from something as I thought, so I, like everybody, I turned to YouTube to try to figure this out, and there was some pretty good videos on how to do it. So thank you to all those people on YouTube who made videos on how to separate DNA. But what you need, is something that something that is easy to extract DNA from. So in this particular example that I did, I used strawberries. Strawberries have large cells in them. Their cells are packed with DNA. They have humans have 2 sets of DNA. Strawberries have 8 sets of DNA per cell. And so lots of DNA to extract. The chemicals and stuff to extract it are pretty easily found. So I had to get pure, 90% or better pure isopropyl alcohol, although I've seen videos where they use like 70% like rubbing alcohol, but I decided to go with pure isopropyl. I got distilled water as the mixing compound for 2 items, which was liquid dishwashing detergent and salt. The dishwashing detergent, I learned today that detergents work by, they have like this, this illustration I saw, it has like this large head that, is attracted to water, and then it has a tail, and the tail of detergent repels water. And so the way that this works, if you're washing your clothes, the detergent tails attract themselves to organics like stains, dirt, that kind of thing. And the head then helps to pull it away from the cloth as you're washing it. So I guess a little chemistry there. Same thing happens with pulling out DNA out of strawberries. The detergent tails insert into the DNA itself. The salt acts as a catalyst to make, it's conductive, and so it helps to pull away the DNA. So that's the extraction process, and then you use cold. Why it has to be cold, I haven't figured that out yet, but I made ice cold isopropyl alcohol. I made isopropyl alcohol ice cold in the freezer, and then I slowly poured it in, and that extracted my DNA into these kind of goopy, almost like snot-like strands. And so I'm going to show you how I did that. I made a little video. So here we go. I left one for me to eat. This is me mixing the detergent and the salt. And now I'm just pulverizing the strawberries in the plastic bag. DNA. Interesting, you just want to pour it in really carefully, like pouring a beer or something. And you can see that almost right away, it starts to separate. This white, kind of white cloud, stringy looking stuff is pure DNA. So I put some of this stuff into a little petri dish that I have. Harold says if I had used double distilled moonshine, it would have made a great cocktail. I noticed that there is a YouTube video. I haven't watched it yet, but they do, they extract the DNA. Now, I imagine that they use the same mixture that I did, but then they eat the DNA. That got 5 million views. I'm not going to eat this stuff, all right? But I'll show you the mixture that I have here, okay? So you can see how it's separated, and I don't know if you can see you can't see down the top, but DNA is still being extracted out of this. And I did this earlier today. I don't know how long it will keep pulling DNA out, but the longer you let it sit, the more kind of pure it looks. Okay, so I did that. I then put my DNA. This is the DNA I have. It's still in the petri dish. And I did pull out some meteorites that I have. I have, different kinds of meteorites. This is Shakota Lin. This is I guess a nickel iron. This one, I can't remember. This is a carbonaceous chondrite meteorite in here. And then this one is Imilac. This is like a palisite, so there would be some crystals inside of it. But those are little samples. Maybe I'll sacrifice one and try to figure out how to extract DNA out of it. I don't know. You'll give me $1 if I eat it. Now, if I could get everybody on Facebook, and I think there's over 1 billion, to send me $1, maybe some fraction of that I would actually eat it. But it's floating in pure 91% pure isopropyl alcohol, so not so cool. So anyways, I decided I prepared a microscope slide, and you can see behind me here, this is my Bresser Infinity microscope. I got my camera up right here, okay, and then I got a little webcam that aims at the microscope. So let me see if I can get this to share the view. I'm not sure if it will. I can see it on Zoom. And it's not. Okay. So what I'll do is I will instead share the view through the microscope itself. And I've put a dark field adapter onto the microscope, replacing this is the regular condenser that fits just right underneath the microscope stage. It does have an iris here, so I can adjust the brightness, but it also affects like depth of field, which is cool. So maybe I can show you what the DNA under the microscope looks like with a dark field and a bright field here. So, let's share my screen. And here we go. So you can see these bright things here. Those are bubbles underneath. These are bubbles. The little bright things are bubbles in between the top of the microscope, the cover slip, and the bottom of the slide itself. But those real kind of hairy, fibrous-looking things is the DNA itself. So while we're not going to see the double helix, I don't have an electron scanning microscope. You can actually demonstrate to students or just to yourself that you can see DNA with the naked eye, and then you can see some of the structure of it at even modest magnifications. I'm going to switch out the adapter here, the dark field for the bright field. Oh wow, look at that. That made contrast a little bit more interesting, just moving it. go. And I put it in the bright field. There we go. And now I have to adjust the exposure time because the dark field, of course, takes a longer exposure. I'll pull that down till we start to see some detail. And there we go. Okay, so now you can see how the view looks this way. And as I change the aperture, you can see also some more interesting effects here. But look how fibrous it looks. So it's pretty interesting. I'm at medium magnification, about 100 power. But that's my Mr. Mad Scientist moment today. So here we are. Let's read some of your comments here. Oh yeah, Chris Larson says it does look rather nebulous. Yeah, that's true. And Norm Hughes says, good afternoon, everybody. Is that the Crab Nebula? Beatrice, how much does the dark field adapter cost that you use now? Does it fit on microscopes other than of other brands or only ones from Bresser? You know, this one, Beatrice, I think works just on the Bresser microscope, but look at how it's made, okay? And I have seen people make their own dark field adapters. It's an occulting disc is what it is, but it's not covering up the entire field, and so light scatters around it and basically gives your specimen edge lighting. So it's good for a lot of microorganisms and stuff like that to see it in some other greater detail. But I like to look at all my specimens in both bright field and dark field. Israel says the best part of MicroCosmos is that it's available when the clouds roll in. That's right. That's true, as they often do here in Arkansas, and they sure do. Andrew says, I need to buy a couple of Bresser Infinity microscopes for a trade show display. Okay, well, that's great, Andrew. We'll talk about it. But I think you'll find that the quality of this particular microscope is really, really good. And it's, I have yet to get some other adapters for it. I'd like to get a polarizing filter, I think. And I have on order, I don't have it yet, but I'm supposed to have a 20 megapixel camera. The one that's on there right now is just a 3.1 megapixel. So anyhow, yes, Beatrice, I think you can DIY a dark field adapter somehow. And someone here from Astronomy Notebook says one can simulate a dark field by using a small dark circular paper. And that's true, you sure can. I remember when I was developing a dark field adapter for a toy microscope, and I was thinking that was really going to be, something very, very specialized, but that's not the case. It really is just an occulting disc. So you can probably make one dark field adapter better than the other, but it does work just to use a circular piece of black paper. And anyhow, that's my presentation for today. It was fun to extract the DNA and to to see that whole experiment work. I plan to do more of that kind of stuff. So So that's the voyage part of MicroCosmos Voyages for me. I hope you guys have a great weekend. Mike Wiesner, I'll give a shout out for him. Again, he is reminding us that there is going to be an asteroid crossing M10, I believe is what it was. So that's something to look forward to tonight, if it's clear. And until later, I guess I'll wish you guys a great weekend, and we'll be back on Monday with Dr. Daniel Barth on How Do You Know? And I wish you all to keep looking up and to enjoy your friends and family as much as you can. Take care. Hey everybody, Scott Roberts here with Mike Hatch, and we're here at Explore Scientific Live, and we wanted to show you the Bresser line of microscopes. Now we're just showing a few of them here. We've got microscopes that do fluorescence work, some used for metallurgy. A microscope like this can look at 3D objects, including things like circuit boards, or if you have insects or anything like that, this does an amazing job. A lot of these microscopes can also take a camera attachment, which is great.
Like these right here with the trinocular?
That's right, and we have a special camera that does that. Some of these other microscopes have a way to attach like your iPhone to them so that you can get images. This one right here has a built-in monitor, so it's pretty cool. The microscopic world is endlessly fascinating, just like astronomy is, and we hope that you take a look.